Hi everyone, my name is Camille Paginto, and today I will be going over the biological technique called RNA interference. So to understand this overall process, we need a little background of what DNA, RNA, and proteins are. So I'm pretty sure we all know that DNA is replicated to make copies, and then transcription uses this DNA to make complementary RNA, and then finally translation uses the mRNA to make proteins. Specifically for RNA interference, the smaller RNAs interfere with gene expression and thus sometimes proteins can't be made. So this takes me to the principle of technique. What exactly is RNA interference? So RNA interference is a process in which the double-strand RNA prevents expression of a gene homologous to part of the RNA. For a little more history background, the process was discovered in 1998 by two scientists named Fire and Mello. They used Caenorhabditis elegans, which is a ringworm, to analyze this process. So they found that the double-strand RNA source of sequence to specific inhibition of protein expression was found in this ringworm. Some key terms important for RNA interference are the short interfering RNAs or siRNA, microRNAs or miRNAs, and these are called dicer enzymes because basically what they do is they dice or cut the dsRNA into smaller fragments. And there's also an important protein known as RISC, which I will talk more of next. This technique can be applied in different scenarios such as identifying genes in a genome that can contribute to disease phenotypes. It can also be used to block an expression of a specific gene, which my figure in the next slide will discuss. And finally, it can be used to assess a response to chemical compounds or a change in a signal pathway. Now the picture on the right over here it shows a picture of the process where we have a double-strand RNA right here, and a dicer enzyme is used to cut the strand into smaller fragments of 21 to 28 base pairs. Now the small RNA and proteins, they will assemble into this purple shape right here. It's called a ribonucleoprotein, where the small interfering RNA is unwounded to make an RNA-induced silencing complex, or the RISC, which I mentioned earlier. So the RISC, what it basically does, it targets a section of the mRNA to make the complementary strand. If it is perfectly base paired, then the mRNA is cleaved and degraded, but if it's not, then the translation basically stops and polypeptide synthesis is not continued. So this figure comes from a research article about a certain gene called YOD1 that encodes an enzyme responsible for non-syndromic cleft lip or with or without cleft palate. The key question that was asked in this figure was whether the detection of YOD1 using RNA interference had an effect on mRNA expression of YOD1. So the experimental design was that they basically used RNA interference on cell proliferation, and they used mRNA levels that were found using a technique called QRT-PCR, which we can see here in B, like the results of that QT-PCR. Western blotting was used to determine the protein levels as shown right here in A. Overall, the data just shows that for siRNA number 3, it is a reduced amount of mRNA and protein levels. So as you can see, it's more faint compared to the others. And here, the levels of mRNA is significantly reduced compared to the others. A strength in this experiment was the use of multiple siRNAs and having replicates. They actually did it 3 times. And a weakness is maybe that the HOK cells, which are the human oral keratinocytes that were used, maybe they were biased or they were not randomized since it was not mentioned in the article. So my question now is, in RNA splicing, we know that the introns are removed and the exons are joined together. So if this did not occur, if the RNA splicing did not occur, will this have an effect on RNA interference? That is all that I have today. Thank you, everyone.